Hello everyone. This session will be about histology of liver and gallbladder. Liver is the second largest organ of the body, as we all know, and it is the largest gland in our body. Located in the right upper quadrant of the abdominal cavity under the diaphragm. The liver also has both exocrine and endocrine function as like pancreas. Here, this is an epithelium, hepatocyte, and histological cross-section of the liver. You can see from outside the mesothelium, and the capsules that covering the liver is the glycine capsules, and in there is hepatocyte and the sinusoid, liver sinusoid. The stroma of the liver is covered by a thin connective tissue capsules. This is called glycine capsules that become thicker at the hilum side. Reticular fiber network support the hepatocyte and sinusoidal endothelial cells of the liver lobules. You can see here from the right side of the screen, this is the reticular fiber that support the hepatocyte and sinusoidal endothelial cells. Here, this picture, you can see the portal triad. The big one is the portal vein and bile duct with the hepatic artery. The liver lobules are divided into three ways: the classical lobule, portal lobule, and liver acines. You can see here the classical lobule, central vein is the center, and in the hexagonal shape, the peripheral is the portal canal, and in the portal lobule, you can see between the three central veins. This is a triangle between the three central veins. This is the portal lobule. Is also the way of dividing the liver lobules. And another one is the liver acines. This is found between two central veins and divided into zone 1, zone 2, and zone 3. We will talk about it. Here is a classical lobule, as you can see. Classical liver lobule. In the center is the central vein, and the corner is the portal triad surrounding the central vein. The central vein, and is hepatocyte and the sinusoid. Here also the classical lobule, you can see the rows of hepatocyte arranged radically around the central vein. This is the classical hepatic lobule. It is very clear. You can see hepatocyte and liver sinusoid, the portal area, portal triad. You can have portal vein, hepatic artery, and bile duct. Very clear. Here also central vein, and you can see hepatic artery and portal vein and bile duct, and also coffer cells and liver macrophage. The portal triad. This is a classical lobule. For the portal lobule, its tissue is found between the three central veins. You can see here also in the left side of the screen, in histological section, you can see the portal lobule. Also here, the branch of portal vein, biliary duct, and branch of hepatic artery. This is the portal triad. This is the portal lobule. And liver acinus. In liver acinus, it's also the space between two central veins. Is divided into zones, starting from zone 3, close to central vein, then zone 2, and zone 1. Here, dividing the portal vein in this way, of the liver dividing like acinus in this way is important when there is a liver fibrosis or cirrhosis, like periportal fibrosis, like in those of the visceral leishmaniasis, like for the egg is lying beside the portal central vein and portal triad of the leishmaniasis, and also in the disease called Buchiari syndrome, and the Pituitary syndrome is periportal fibrosis. Also, fibrosis starting from the zone 3, 2, and 1, and the seriousness of the fibrosis when it's close to the central vein, as then causing ascites, everything like that. Hepatocytes. Hepatocytes are the large, this is the liver cells, hepatocytes, the liver cells, are the large and biogonal cells. They constitute about 80% of the cell population of the liver, arranged into plates. Hepatocyte nuclei are large and spherical, occupy the central cells. Many cells in the adult liver are binucleated, and most cells in the adult liver are tetraploid, and they contain 4 types of the DNA. Contain numerous also mitochondria, as many as like 800 to 1000. Here, this is liver hepatocytes, liver cells. You can see all cell organelles are there: reticulum, smooth reticulum, mitochondria, Golgi, collagen fibers, and also macrophage around site, collagen fibers. Liver, sinusoid, and space of this. Hepatic sinusoid are lined with the characteristic type of epithelium is called discontinuous endothelium. And sinusoid of the liver cells are characteristically fenestrated. They have pores or fenestrated. Hepatic sinusoid is different from other sinusoid in that contain second cell type is called sinusoidal macrophage. A special type of macrophage is called Kupffer cell of the liver. In the perisinusoidal space of this lies between the hepatocyte and endothelial cells. This is the perisinusoidal space of this between the hepatocyte and endothelial cells, and Kupffer cells that lie in the sinusoid. In the fetal liver, a space of this contain island of blood-forming cells. In the case of chronic anemia in adult, blood-forming cells may again appear in the perisinusoidal space. This is important of space of this. It's also this contain cells that store vitamin A. In certain pathological conditions such as liver cirrhosis, hepatic stellate cells lose their lipid and vitamin A storage capacity and differentiate into myofibroblasm. That differentiation will lead to the cirrhosis. That's the pathology of cirrhosis. Here again, this is the hepatocyte. You can see here cells in the perisinusoidal space between the hepatocytes and endothelial cells. This is a perisinusoidal space, and that one containing the RBC is the lumen of the hepatic sinusoid, as you can see clear here. Hepatocyte produced bile which is secreted into, this is a bile flow or bile canaliculi, passage of bile from hepatocyte into bile canaliculi into the hepatic lobule, and after that into the bile duct, and then differentiate into the right and left hepatic duct. This is a flow of bile from hepatocyte. You can see here from bile canaliculi, from the hepatocyte itself, the bile canaliculi, and then to the bile ductule. After that into the bile duct, and then to the right and left hepatic duct. Let us talk briefly about gallbladder histology. The wall of the gallbladder consists of a mucosa composed of simple columnar epithelium. This is the most important type. This is the mucosa of the gallbladder is lined by simple columnar epithelium. Important. And also have lamina propria and sine muscularis and interna and externa, and also adventitia. The mucosa has numerous folds. That's the characteristic of gallbladder. Mucosa has numerous folds that are particularly evident when gallbladder is empty. That can be seen obviously when gallbladder is empty. The deep diverticula of the mucosa is called rectangulate sky Ascoff sinuses. Sometimes extend through the muscularis externa. This is the diverticula of the mucosa of gallbladder. It is called Ascoff sinuses, which you can extend into even to the muscularis externa. Let's see into the next picture. Hepatocytes. Hepatocytes are the large, this is the liver cells, hepatocytes, the liver cells, are the large and biogonal cells. They constitute about 80% of the cell population of the liver, arranged into plates. Hepatocyte nuclei are large and spherical, occupy the central cells. Many cells in the adult liver are binucleated, and most cells